Let's talk about the mitotic phase. The mitotic phase is made of two parts. The first part is nuclear division. That's when the nuclear material divides into half and moves to the two ends of the cell. This is something that is called mitosis. That's the process that is called mitosis. Followed by that is cytoplasmic division. That's when actually you're going to end up with two cells at the end of this stage. Let's take a look at a video of this process. So here it is. This is chromosomes are becoming condensed and they're trying to line up at the middle of the cell. So there's lots of pulling and pushing going on. And you will see at some point all of the chromosomes are going to go to the center of the cell, which what we call the metaphase plate. So all these chromosomes are all going to line up in here in the middle. And then at a snap of a finger, they all separate. So now you get one set of genetic material in this end, some set of genetic material in that end, and then finally you get cytoplasmic division and you get two cells. So here it is. These are the microtubules. They're like ropes that are going to pull the electrons away from one another. So here it is. They're lining up at the center and being pulled apart. So these are microtubules. There's like ropes that attach to the centromere and pull the chromosomes apart. So this happens in various different stages, and we're gonna go over each stage. Let's talk about the different stages. There are multiple stages in mitosis, and we're gonna concern ourselves with the few major ones. The first stage right after the G2 phase is called prophase, and these are the events that happen. But first, let's take a look at the cell. So there is the nuclear membrane, the chromosomes are condensed, and then these structures, the orange structures are centrosomes, and is emanated from them these long ropes of protein, which are called microtubules. So during the prophase, chromosomes condense and become visible. Spindle fibers emerge from the centrosome. So these blue lines are the spindle fibers. Nuclear membrane starts breaking down, and centrosomes move toward the opposite poles of the cell. These two. During pro-metaphase, chromosomes continue to condense. Kinetochore appears at the centromere. I'll show you a close-up of the kinetochore. Kinetochore is basically a complex of proteins that establish themselves at the chromosomes. And then mitotic spindle, which are the same as spindle fibers. Mitotic spindle microtubules attach to the kinetochore. So here's an artist's rendition of a kinetochore. This is a chromosome, this purple structure. Let me bring my laser pointer. This structure is the chromosome. This is the centromere. Kinetochore is a protein complex that attaches itself to the centromere. And then this kinetochore holds hands, takes like a grip of the microtubules. Microtubules are those long proteins that we talked about when we talked about the cytoskeleton. So these attach themselves to the centromere. And this is how, when chromosomes line up, this is how you can separate the sister chromatids, because these are like ropes that are pulling things toward them. So these fibers, microtubule fibers, are going to pull the chromosomes towards the two ends of this cell when anaphase comes, and we haven't talked about that yet. Okay. So another thing I wanted to point out is that by the time the pro-metaphase comes in, the nuclear membrane totally disappears. So first, chromosomes condense, become visible. During the prophase, the centrosomes start emanating, getting out these spindle fibers, and they move towards the two ends of the cell. And the nuclear membrane starts breaking down. By the time you get to pro-metaphase, the chromosomes are starting to attach through the kinetochore to these fibers. Then metaphase is that place where everything, all the chromosomes line up at the center of the cell. So chromosomes are lined up at the metaphase plate. The center of the cell is called the metaphase plate. And each sister chromatid is attached to a spindle fiber originating from opposite poles. So you can think about this. You have these sister chromatids that are attached through the centromeres. One of them is being pulled attached to this side, and the other one is attached to ropes on the other side. During anaphase, these ropes start pulling. They start pulling the chromosomes away from one another. So centromeres split in two. Sister chromatids, which are now called chromosomes because now on their own right, they are no longer anybody's sister. They're their own chromosomes. They are pulled toward opposite poles. And then certain spindle fibers begin to elongate the cell. Okay? So sister chromatids were attached here together. Here they're separate. During the telophase, you start building the nuclear envelope back. So chromosomes arrive at opposite poles and begin to decondense. Nuclear envelope material surrounds each set of chromosomes, and the mitotic spindle breaks down. So mitotic spindle is not needed anymore, so it starts to break down. But they continue to push the cell towards the two ends. So that is the end of the the process called mitosis. Then comes the cytokinesis. Before we go there, I wanted to show you another image. So here's another. It's a GIF of cell division. So you can see chromosomes condense, come together, they go to the center, and they get separated. So mitosis is over. Now we're going to go to cytokinesis. Now cytokinesis happens in two different ways if you're looking at an animal or a plant cell. During the animal cell, towards the end of telophase, which is the end stage of mitosis, a contractile ring, which is made of actin, these microfilaments form here, and then they start putting a lot of pressure on this area of the cell between the two newly formed nuclei and form what's called the cleavage furrow, and they keep stretching and stretching until the two cells snap apart. That's how it happens in animal cells. In plant cells, it's different. There is no cleavage furrow or tension in here. What happens is that there are Golgi vesicles that move and position themselves in between the new nuclei, and they start producing material that would make the future plasma membrane and cell wall. And then the two cells divide. So stretching in here and putting stress between the two half of the cells here and here, there is no putting stress. You just actually build a wall from scratch. So there are a couple of questions that I have in here.